So our first spotting question is showing you a cell. Okay, so it's a cell. We know this is the nucleus, this is the cytoplasm or the cytosol. Okay, so obviously it's a cell, showing you some steps that are taking place in the nucleus. Okay, so in the spotting there could be some blank areas. You have to name those things. Okay, you might have to name those things, the steps taking place, or the product that is being formed. Okay, or you might be asked about what is this picture depicting you. Okay, so obviously let's name the parts of the cells. So the outer border of the cell, we known as, we say those to be the plasma membrane. Okay, or the cell membrane. Okay, this is the nucleus. Obviously this is the nucleus, so the membrane that is covering the nucleus is called as the nuclear envelope or nuclear membrane. Okay, so inside the nucleus, we know the genomic sequencing are there, kept in a strand, highly compact strand called as the DNA, which is actually the full form of deoxyribonucleic acid. Okay, so the D something from the double helix DNA, a single strand of genome is being produced, which is called as RNA. Okay, the full form of RNA is ribonucleic acid. Now the step which helps in forming RNA from a deoxyribonucleic acid is called as DNA transcription. Whenever from a DNA RNA is formed, is called as transcription, SCRIP. Okay, now inside the nucleus there are some enzymes are there which helps in cutting out some segments of the RNA which is not required for the further step. Okay, from this sequence only we can produce, our body produce some proteins. Now for that sequencing, for the production, some specific genomic sequencing is required. Okay, so the splicing occurs. Okay, this is called as splicing, cutting out of the extra extrons. Okay, which is called as extrons. So then we get this extrons with the help of an enzyme called as RNA splicer, and which this process is called as RNA splicing. The splicer helps in doing RNA splicing, and we get a strand of single strand of RNA which is called as mRNA or messenger RNA. The RNA, the name, it means ribonucleic acid. Now the mRNA that is formed, it is formed inside the nucleus. Now something has to be formed inside the cell. For that production of protein, it has to be done in the cytosol. So we need some vehicles. Okay, there are some transport proteins are there which helps in transporting this mRNA. So these are called as the RNA transporters, which helps in this process. This is called as RNA transporting. Okay, so the RNA transporters helps in transporting the mRNA to the cytosol. Now inside the cytosol, so there are some organelles, 60S, 70S, 80S, ribosomes. Okay, now this enzyme, these organelles or this specific proteins helps in the further steps of your mRNA modification, in which the mRNA is helped by the ribosomes to form a strand of proteins. Okay, these are the primary strands of proteins that are being formed. Okay, now the step of formation of something of protein from an mRNA is called as translation of mRNA. The messenger RNA, there are some codes there inside the mRNA strand which helps in translating SLA, translating the sequence into a protein. Okay, so basically in the picture that is showing on the right is showing you the flowchart mechanism that is taking place in the cell. So from the nucleus, the genes containing the DNA, deoxyribonucleic acid, with the help of transcription pathway, it forms RNA. Okay, RNA is formed. Now this RNA then goes through a process in the cytosol, okay, called as translation, and it forms the protein. Okay, now these proteins do some specific cell structure modification as well as production of some enzymes. Okay, so modification of the cells based upon the enzyme formation or the structural framework of the cells of the designated or destined target cells, it modifies the cellular function, and thus the gene works in our body, okay, in a cellular level. So this figure is showing you the general scheme by which genes control cell function. Okay, now here this is the picture's description. Now what is this picture showing you over here? Okay, so here you can see a bilaminar structure is there, having a globular end and two arms are there. These are called as the lipid layer. Okay, lipid bilayer. So we have two layers there. So this layer is showing you, this whole layer is showing you the cell membrane. Okay, so cell membrane, it's a bilaminar lipid layer having a mosaic appearance. So you know what's a mosaic appearance. So there are some inside the granular structures, there are some big and some small circles and some big structures are there. So these are a mosaic. This all the flooring patterns that are you find in your home, that's called a mosaic. So the mosaic pattern is present in the cell membrane also. So it is formed by the lipid layers and some proteins. Okay, so there are some proteins that is passing through and through. Those are called as transmembrane proteins. So these are all passing through and throughout the layers of the lipid bilayer. So these are all transmembrane proteins. There are some proteins which are, so by the way, the area is called as this is outside, this is called as extracellular fluid. Okay, extracellular fluid. Okay, and this part which is inside the cell is called an intracellular fluid layer. Now there are some proteins that you will find in the cells which are passing through and throughout. These are called as the transmembrane proteins. Transmembrane proteins. Okay, that is passing through and throughout the membrane of the cell. You also will find some proteins which are sticking to the outer edges. These are called as extracellular proteins. Okay, and some proteins that you will find attached to the intracellular layer. These are called as the intracellular proteins. Okay, now because of this mashed up appearance of transmembrane proteins, extracellular proteins, intracellular, it gives the appearance of a mosaic. Also, you will find some structures which are attached to lipid membrane. Take for example, if you find a carbohydrate moiety or carbohydrate molecule sticking to the or combining or forming bond with the lipid membrane, okay, outside, is called as glycolipid molecule. Supposedly it is not attached to the lipid, but it is attached to either the transmembrane protein or an extracellular protein. Those molecule is called as glycoproteins. Okay, so these are the cellular structures which gives the appearance of fluid mosaic model of cell membrane. Fluid mosaic model. Okay, so Singer and Nicholson was the two physiologists who discovered for the first time this fluidic mosaic appearance of the cell membrane. The mosaic is because of the patterns or the molecules that are present in the cell membrane, and fluidic nature is because of the constant movement of these molecules. So these structure that you can see, these are not always stagnant. Every second of your life, every cells of your body have constant motion of all these molecules, glycolipids, glycoproteins, the lipid membrane, the transmembrane proteins, all things are in a constant motion. So it gives the appearance of a fluid which is in a constant motion. So Singer and Nicholson found those layers to be constantly moving, and the movement was in a mosaic kind of pattern. So that's why they gave the name as the fluid mosaic model. Now this was about the structural details. Now let's see what are the different transmembrane proteins that you will get. Okay, now a protein you will get, so basically there are some substances which needs to be transported from inside to outside, and some substances which need to be transported from outside to inside. So the cell needs some mechanics or some gateways to transport those substances. So in our cell we have certain transmembrane protein which are called as the channel proteins. Channel proteins, okay, which transports some substances from outside to inside or from inside to outside with the help of this protein. So this protein, you can think it to be like a tunnel, okay, which is connecting two edges of the cell, two sides of the cells. Okay, there are other transmembrane protein, okay, which also transports the substances from inside to outside or outside to inside. Okay, but in this case they are usually interacting with the transmembrane protein molecules. Okay, so those are called as your carrier proteins. Okay, carrier proteins. Okay, now there are some carrier proteins which does bidirectional function, and there are some carrier proteins which does unidirectional function. Okay, the difference between these carrier proteins, this and this, is that there are certain carrier proteins which requires energy for transport of the molecules. Okay, and some carrier proteins which does not need energy, no energy is needed. Now both of these three together, carrier proteins, are helping in transport of the substance. Okay, the one which is using the energy are called as active transport. Okay, so actively it is using the energy, and that's why it's called as active transport mechanism of transport of the substances. Okay, on the other hand, this carrier proteins which is helping in transporting the substances with interaction of the channel proteins, carrier protein molecules without the using of energy is called as the facilitated diffusion. Okay, whereas this channel proteins, which is transporting simply the substances from outside to inside or inside to outside without interacting with the molecules of the protein, okay, just acting as a tunnel, is called as a simple diffusion. No energy, no interaction, so that's why it's called a simple diffusion. So this phase of diffusion and this phase of diffusion with the help of a carrier protein without using the energy is called as diffusion. Okay, so diffusion, you can think it to be that part of the transport which does not uses energy. So there are two types of diffusion. One is called a simple diffusion, another is called as a facilitated diffusion. Simple diffusion uses the channel proteins, and facilitated uses no energy but uses a carrier protein. Okay, but with the interaction of the molecules that is present in the protein. Okay, another important point to note down in the carrier protein transport is that apart from using energy, okay, when the substance is attached or interacting with a part of the carrier protein, it leads to change in conformation. Okay, so that's a very important conformational changes. Okay, so there is a conformational change in the structure of the carrier proteins. So if this is a cell having a typical morphology of supposedly a cell, okay, when it attaches to a molecule which needs to be transported, it then changes in its conformation, okay, to a something which leads to release of the molecules to the outside. Okay, so like this it changes the conformation. So interaction, energy, and change in conformation. Now active transport active transport process is again of two types. One is the primary, another is the secondary. Okay, both of these mechanisms use energy, the primary mechanism and the secondary mechanism. Now what happens in the primary is that the molecules which are being transported, okay, will be either transported singly or would be transported with the help of a substance called as ATP. Okay, since it's using the energy, so ATP has to be used. Okay, so the ATP is directly broken down. Okay, is broken down, and the energy that is released, it helps in releasing the substances from either from outside to inside to or from inside to outside. Okay, the perfect example for primary transport is sodium potassium ATPase pump. Okay, in any pump, if it is written ATPase pump, so those pumps are primary active transport mechanism pump. Okay, so what happens in that pump is that the sodium potassium ATPase pump, supposedly this is a carrier protein called as sodium potassium ATPase pump. There we have some sites on the extracellular fluid and the intracellular fluid. Okay, in the intracellular fluid there we have a sodium channel site, sodium ion site, and for the extracellular the potassium site is there. Now in the intracellular there are a provision for attaching to 3 sodium ions, whereas on the extracellular level there were 2 potassium ion attaching site. Along with that we have a site which attaches the ATP molecule. So there is a ATPase enzyme over here which breaks down the energy, and this energy is used to change the conformation of this molecule. Okay, and with the help of that, what happens, the molecule transports the substance, the potassium will go inside and the sodium will go outside. Okay, whereas in case of secondary active transport, what happens, although the energy is used to change the conformation of the carrier protein, but there is a difference between the primary and the secondary. What happens is that supposedly this is a carrier protein which is transporting a substance from outside to inside to inside to outside. What happens is that there are two basic types of secondary transport mechanism. Is that one in which it is transporting the substances, not one or two. Okay, so if supposedly there is a molecule A and molecule B, it can either transport it together, or there are certain carrier proteins which helps in transporting the substance in bidirectional direction. Okay, so if it is A from outside to inside, B from outside. So the left picture is showing you the co-transport mechanism of secondary active transport, and the right picture is showing you counter. So there is a counter direction. Okay, counter direction, counter transport of secondary active transport. Now here also the energy is used. Now the trick part over here is that here, if supposedly the energy or the ATPase molecule is been broken down, okay, to transport supposedly substance A from outside to inside, the energy that is released will help in movement of the other substance from inside to outside or outside to inside based upon counter or co-transport mechanism. So one substance is transported with the help of energy, and this energy is then used for transporting the second substance. Or you can say, if one substance is being transported because of the concentration gradient or chemical gradient, the second substance will use the energy. So one substance will use the energy, other substance won't use the energy. The perfect example for the secondary active transport co-transport is sodium glucose co-transporters that is present in the GIT. Okay, the perfect example of the secondary active transport are sodium chloride transporters, okay, or sodium hydrogen transporters, okay, or sodium magnesium transporter, or calcium magnesium or potassium transporters. Okay, so our figure is showing over here is the transport pathway through cell membrane of cell, and the basic mechanisms of transport. Now next picture is showing you what. Okay, so the structure that you can see over here is a neuron. Okay, it's a neuron, so the functioning unit of a nerve. Okay, so the neuron, you know, this is the cell body, okay, having the nucleus and the cytoplasm or the cytosol. The cytosol is connected to a filamentous strand on which some structures are present layering over the filament. Okay, so this is called as axon. Okay, and the filaments which are coming or emerging from the cell body is called as dendrites. DENDRITES. Okay, so usually the signal that is transmitted in the nerves, it is arriving with the help of the dendrites. It is passing downhill along the axon. Okay, so whatever action potential is generated in the axon, it starts at this particular point. Okay, so this is the point at which it is called as the action potential starts over here. Okay, so the layer that you can see, which is transcribed, it has been shown very clearly in this picture. You can see there are layers of lipid membrane is there. Okay, so these are called lipid membranes, which is called as myelin sheet. Okay, these are called as myelin sheet. Okay, now you can see there are some areas in the myelin sheet where there is no, in the axon where there is no myelin sheet present. Okay, so these areas are called as, these areas having no myelin sheet is called as nodes of Ranvier. R-A-N-V-I-E-R. Okay, Ranvier. So inside this Ranvier, you can see there are some channels which are helping some ions to pass through. Like for this example, in the nerve, the sodium ions are being transported from outside to inside. Okay, so since most common cation in the extracellular fluid is sodium, so it's been shown over here. Now in the nerves, the spatial propagation of the action potential that is arriving in the cell body, okay, through the by the help of the dendrites, it starts from the junction of the cell body and the axon. Okay, it jumps from one nodes of Ranvier to the other nodes of Ranvier. So this typical movement of the action potential that is arriving, this is action potential, it is jumping from one nodes of Ranvier to another, is called as saltatory conduction of electrical signals, which takes place in the nerves. Now why this is happening in nerves is because in the nodes of Ranvier, what happens, all the ions are getting accumulated. Now what happens, for passing or for depolarizing the part of the exposed axon, it requires few ions which will depolarize this section of the neuron. Now what will happen to the rest of the ions, it will then pass along the nodes of the Ranvier, myelin sheet, and it will go and directly depolarize this section of the exposed nodes of Ranvier. So why this saltatory conduction is important in our nerves? Because it helps first in fast conduction of impulses in the nerves, which is the basic necessity for our reflexes and day-to-day life activities that we conduct with the help of the muscles. Okay, also it requires less ions to depolarize. Okay, so few amount of ions are going from here to here. Okay, remaining which is there will again go to the next part. So that's why this conduction is important. So this picture showing over here is saltatory conduction along a myelinated axon. Okay, dissect this area and look closely under the microscope, you will be able to see the eye band. This eye band is this area. Now I told you inside the eye band there is a light zone area. This light zone area, it is called as age zone. Okay, age zone. And on the either side of the age zone we have the dark band area. Okay, and either side of the dark band area, this is the eye band area. Okay, this is the eye band area. This is the eye band. This eye band is shown over here, and this eye band is shown over here. Okay, this is the eye band. Now either side of the eye band, I told you there is a Z line, so this is the Z line shown over here. This is showing you the Z line. This is also showing you the Z line. Now in between these two Z lines, that is this one particular Z line and this particular Z line, we call that as the length of the sarcomere. Okay, so if we again go much deeper in the visualization of the sarcomere, you will get the A band looks like a combination of some thick strands and some thin strands. Okay, now the thick strands and thin strands, these all together are called as myofilaments. Okay, myofilaments. So individually the myofilaments, if you look into this myofilaments, if you enlarge these myofilaments, you will see it's a collection of hockey stick shaped some filaments. Those are called as myosin filaments. Okay, along with that the thin strands that you can see, okay, this thin strands you can see, these are called as actin filaments. Okay, along with that actin filaments, you can see some globular molecules, protein molecules are there, so those are called as the G actin molecules. Okay, and this strand is called as the F actin molecule. Now if you go much deeper into the layer, if you take out one hockey stick filament, okay, this is called as myosin molecule. Okay, this is called as myosin molecule, MYOSIN. Now the myosin molecule is made of a tail, it has a hinge, and it has a head. Okay, and this picture is showing you the cross-sectional morphology of the strand. If you take a cross section at this particular section, you will see these small dots, they are showing you the F actin molecules. If you're taking a cross section here, you will be able to see the H zone myosin tail. If you're taking a cross section from here, you will be able to see is the, along with the myosin tail and F actin, you will be able to see the hinges also. Okay, so these are the section which is showing you the organization of the muscles in the human body. Specifically, it's a skeletal muscle organization, okay, at the molecular level. Now what is this picture showing in figure A and figure B? Now figure A is showing you a myosin molecule. Okay, so this is a myosin molecule having two distinct parts. One, a strand looking part which is called as tail, and a globular, more or less a globular part which is called as head of the myosin molecule. Now inside the head there are the strands of protein molecules in entangled manner. Okay, and along with that we have a light chain, and these are the heavy chains. Okay, so this is a myosin molecule. Now what is shown over here is in the B is that a picture of many such myosin molecules. The hockey stick like picture you can see at the very center, these are nothing but the myosin molecule. So this is one myosin molecule, this is one other myosin molecule. Like this, all many myosin molecules are present in the myosin filament. Okay, so it is showing you a myosin filament having many such myosin molecules. Okay, so this is the head part of the myosin molecule and this is the tail part of the myosin molecule, and this part is called as the hinge. Okay, the connection between the tail and the head. Okay, so this, what is this? This is a typical actin filament. Now in a muscle myofibril, you know, the muscle tissue, the myosin filament is made of actin and myosin. Now when we do contraction, there should be a process that is generated called as the cross bridging. Okay, or sliding filament. Okay, what happens, the myosin is attached to the myosin binding portion of the side of the actin molecule, which drags the muscle either towards the center or the sarcomere or the Z line, or away from the Z line. Okay, depending on whether contraction or relaxation is required. Now this is the B, which is showing you a cross bridge. Okay, BRIDGE. So there is a bridge being formed between the myosin molecule and the actin molecule. So here for example, these are the cross bridges. Okay, so B over here is showing you combination of many myosin molecules to form a myosin filament. Also it is showing you the cross bridging, okay, between the heads of the myosin and the actin molecules. So in this picture there are three figures given over here, A, B and C. What is being shown over here? Okay, so A is showing you, now all these figures are showing you the different views of motor and plate. Okay, so it is showing you the motor end plate. A specifically is showing you the longitudinal section, longitudinal sectioning of the motor end plate. Okay, B is showing you the surface view, and C is showing you the electron microscopic view of the motor end plate. So basically this is the junction between the muscle and the nerve. Now in the A figure, this part is showing you the muscle. Okay, this part is showing you the myofibrils. Okay, the collection of actin and myosins are there. Okay, many such filaments are combining together and forming a fibril. Now this is the nucleus. These are the nucleus of the muscle tissue. This is showing you the teloglial cells. So these are the nursing cell, or these cells provides nourishment to the muscle tissue. Okay, now this over here is showing you the end of the axon. Okay, and you know, usually the nerves are if a myelinated nerve is there, we have a layer of myelin sheet. So this is nothing but a myelin sheet. Okay, and this is nothing but the terminal portion of the nerve. B is showing you the surface view, in which this is the nucleus of the muscle, and these are the terminal portions of your nerve, and these are the muscle tissues or myofilaments. Now in the electron microscopic view, you can see a typical muscle and a axon terminal is there. Okay, if you look closely, supposedly if you are enlarging this portion, the terminal globular portion is called as the terminal portion of the nerve, which forms a knob like structure, is called as synaptic knob. This whole structure is called as synaptic knob or axon terminal or synaptic knob, knob. Okay, so this is a myofibril, okay, having a downward depression. So this downward depression is called as synaptic trog. Now inside this synaptic knob, there are some vesicles are there which contains the neurotransmitters, which are called as synaptic vesicles. Okay, and the rugae that is formed in the synaptic trog to increase the surface area of this myofibril is called as subneural, that is below the neural tissue or the nerve cleft. Okay, so the figure over here is showing you the different views of the motor end plate. So these two charts are given. So the upper picture is showing you the action potential in nerve, and this is showing you the action potential in skeletal muscle. Okay, both of these are showing in the x-axis the time duration, and in the y-axis both of these is showing you the amplitude of the potential in millivolt. Okay, the difference between these two, although it looks same, it's basically on the resting membrane potential. Okay, the levels at which it is achieving the firing level or the threshold level and the depolarization. Okay, in case of your nerve, the nerve are slightly less negative as compared to that of the muscle. The resting membrane potential of the nerve is 70 minus 70 millivolt. In the case of the muscle, skeletal muscle, is minus 90, so it is much more negatively charged than membranes, whereas the nerves are slightly easily excited, or it is a little bit excitable as compared to the skeletal muscle. Then as soon as we are conducting the experiment, okay, obviously we attach electrodes to give electrical shock, and then we detect with a sensor. Okay, supposedly this is the point at which you are giving the electrical shock. In both of these you will get the stimulus artifact. Okay, now as soon as the stimulus artifact is achieved, in the curve we will get a jerky curve in the action potential curve. Okay, then the curve will be flattened for few millisecond. Okay, then the ions of the extracellular fluid, the sodium ions will try to get inside. First few sodium ions will come inside the channels and you will get slight upward curve. Okay, so it will keep on moving till it reaches a threshold of minus 55 millivolt in case of the nerves. In case of the muscle, minus 75 millivolt. So this is the least volt that is required in nerve and the muscle in order to achieve the action potential. If a electrical shock or stimulus is not achieving this least minimum threshold volt, it won't lead to action potential. So as soon as it crosses this threshold of firing level, we get a depolarization curve. Okay, in both of this curve you will get a depolarization curve. So this is called as depolarizer phase of the curve. Now as soon as it overshoots the 0 millivolt marking, this is called as overshooting part of the depolarization curve or the action potential. Now as soon as this depolarization is occurring because of the opening of all the sodium channels, fast acting sodium channels. Now as soon as it reaches the peak, in case of the nerve it is at the level of plus 35 millivolt, whereas in case of the skeletal muscle the peak is achieved or the spike potential is achieved at plus 55 millivolt. Okay, now as soon as it reaches this volt, the sodium channels starts to close and the depolarization phase starts. So the depolarization phase is starting because of the closure of the sodium channel as well as opening up of the potassium channels. So the potassium ions will flux and we will get gradually the depolarization curve. Okay, now the potassium ion channels are slow to react to open as well as to close. Now as soon as it is starting to open, it also requires some time to close. So because of that, when it reaches the level of resting membrane potential, that is minus 70 in case of the nerve, it crosses up till it reaches a point at which the potassium ion channel closes completely. It's around minus 90 to minus 95 millivolt, after which it again goes back to the baseline. Now this part that is shown over here, this curve, this is called as after hyperpolarization. So up till this it was the repolarization, and after this is called as after hyperpolarization. Okay, in case of the muscles, skeletal muscle, the repolarization when it's taking place, when it reaches the baseline, there is a minus 90 millivolt, it overshoots. Okay, this is called as after hyperpolarization. Okay, now this picture is showing you the action potential in smooth muscles. Okay, this is showing you the action potential in smooth muscle uterus, and this is showing you the action potential in cardiac muscle. Okay, now this is a typical action potential curve of a smooth muscle, showing you the latent period. This is the latent period. Okay, this is the depolarization phase, and this is the repolarization, and again the resting membrane potential. So the depolarization is because of the opening of the sodium channels, and repolarization is because of closure of the sodium channels and opening of the potassium ion channels. Now specifically this particular curve that you can see is showing you the action potential in visceral or intestinal smooth muscles. Now the difference between the action potential in other smooth muscles and the intestinal smooth muscle is that there is a typical undulating resting membrane potential. Okay, this is called as the BER, that is basal electrical rhythms. Okay, so this is called as basal electrical rhythms. Now these are always present in the gastrointestinal tract, so every now and then there is some food particles or the enteric nervous system stimulates the part of the section of the GIT, and whenever the part is stimulated and crosses the threshold, so whenever it is stimulated, specifically the areas that is near the intestinal cells of Cajal, intestinal cells of Cajal, what happens, there is generation of some spikes over the BER. Okay, so these are called, this leads to mechanical events like peristalsis or migrating motor complexes. Okay, on the other hand, in uterine smooth muscles there is presence of some slow channels also. Okay, now the smooth muscles or the skeletal muscle action potential we saw there, we have the fast sodium channels which opens quickly and closes quickly. But in the uterine smooth muscle, what happens, apart from having the fast sodium channels, it also has slowly acting calcium channels. Okay, because of which we get a plateau phase. Okay, so we have a phase 0, phase 1, then we have a plateau phase 2. Okay, and then we have a downward slope called as repolarization phase, and we have a resting membrane potential. Okay, so phase 0 is because of the depolarization of the sodium channels, and phase 1 it reaches the peak from at which the sodium channels is closing and the slow channel calcium channels opens. It takes some time to close, so because of that, what happens, along with that also the potassium ion channels is opening. Okay, now potassium ion channel is opening and calcium ion is opening, so potassium channel is sending the ions from inside to outside, and calcium is sending the ions from outside to inside, so there is a plateau phase. Now after this, what happens, the potassium ion channel is still open, but the calcium ion channel it closes, so because of that we have a downward slope which is called as the repolarization phase. Now in case of the action potential in the cardiac muscle, we'll have the similar nature of the curve as we got in the uterus, is that we have a 0 phase which is because of the depolarization phase of the opening of the fast acting sodium channels. The phase 1 is because of your action potential reaching the peak at which the sodium channels closes. Okay, sodium channel, it closes, but what happens along with that, the potassium channel opens, now which sends the potassium ions from inside to outside. Along with that, the slow calcium channels are also opening. Okay, so which sends this ions from outside to inside of the cell. Now because of that, we get a Plateau phase. And at this point, what happens, the sodium channels are closed, the calcium channels also get closed, but the potassium channels are open. So because of that, we get a downward slope, which is called as the phase 3, which is also called as repolarization phase. Okay, so 0 is showing you depolarization phase, A is showing you the peak or apex or spike potential, 2 is showing you the plateau phase, and 3 is showing you the repolarization phase, and 4 is showing you the resting membrane potential. So the below diagram is showing you the relative point at which the channels are opening. As I've told you, the phase 0 is showing you the fast sodium channel opening. So see, the fast sodium channel picks up. Okay, there is an inward influx of the ions in the sodium channel. At this point, the calciums are starting to open up from the phase 1. Okay, then it reaches a peak. Simultaneously, the potassium ion also picks up, but this ion is taking this channel is taking the ions outside. Okay, that's the action potential for you in case of the smooth muscle and cardiac muscle. Now remember, this is the action potential of a cardiac muscle. Okay, general cardiac muscle, not the pacemaker cells, okay, not the pacemaker cells. Now comparing the last curve, that is the action potential in cardiac muscle, especially the ventricular muscles, are looking like this. Okay, this picture over here, this and this, is showing you the action potential in cardiac tissue, especially of the pacemaker cells. Okay, so at the right side of this chart, you can see there is another curve drawn over here. This is nothing but the curve which is shown over here. Okay, so now we have these two pictures that learn what is the difference between the same organ having the ventricular muscle action potential look like this, whereas in case of sinoatrial or AV nodal fibers tissue, you get the action potential in a sloping manner. Okay, the first difference is that the resting membrane potential of the ventricular muscle is around -90 millivolt. Okay, as compared to that of the nodal fibers, the resting membrane potential is around -55 millivolt. Okay, now why this is the case in case of the sinoatrial node is that in case of sinoatrial node, the fibers, the cells have some leaky sodium and calcium channels, so which opens up and transmits or transports the ions from outside to inside. So there is, at the beginning only, you will get this lesser resting membrane potential. Now this resting membrane potential is -55. Now remember, from this curve I told you, as soon as the channels open, okay, is stimulated, the fibers are stimulated, it opens the fast sodium channels, right? Now because of that, we get this upward slope. Okay, because of the fast sodium channels, this. Okay, now there's another point, that fast sodium channels are fast to open, as well as fast to close also. Okay, quickly it closes also. So usually it has been seen that around minus 55 millivolt, what happens, the fast sodium channels closes. Okay, most of the 90% of the fast sodium channel closes. Now because of that, what happens, although there is slight bit of slope being generated by these fast sodium channels, but because of the immediate closure at this particular point of the firing membrane, firing level, what happens, the slow L type calcium channels come into play. Okay, they are very slow to react or open. Okay, so that's why we get a slope. Okay, we get a slope. Now this slope is achieved because it takes up time to reach the peak. Okay, as it reaches the peak like this, you will get a steeper curve. So at this point you will get, it crosses the minus 40 millivolt, and you get this is the firing level or the threshold for discharge, and you get an action potential or the depolarization phase of the action potential of the cardiac pacemaker cell. Now as soon as it reaches the 0 or plus 10 millivolt, what happens, the cardiac slow acting calcium channels start to close. Okay, it start to close. Now they are not immediately closed. Okay, all the 100 if 100 calcium L type channels are open, all the channels are immediately not closed. So what happens, some 50% closes first half of the repolation, then next 20%, next 10%, like this gradually closes. So because of that we get a downward slope. Okay, we get a downward slope. Okay, having a widened angle. Okay, or widened slope. Along with that we have the potassium channels, which also opens up. Okay, it opens up. Okay, it transmits the potassium ion from inside to outside. Okay, and the calcium channel, it starts to close. Okay, slowly. So as soon as it reaches this point, that is minus 55 millivolt, all the calcium ions channels are closed. But simultaneously, I told you, as soon as it reaches minus 55, then minus 56, what happens, the sodium channels, the fast sodiums again come into play. So again it's depolarizes. So immediately the L type sodium channels, slow calcium channels again starts to open. So like this, there is a complete refractory period in the whole sinus nodal fibers. So you get a long refractory period, because immediately after it is repolarized, it is again starts to depolarize. So this picture is showing you how a immunoglobulin molecule looks like. Okay, so immunoglobulin molecule consists of heavy chains. Okay, it's a Y-shaped molecule having a protein which is called as heavy chain, and we have a protein which forms the arms of the Y called as a light chain. Now this is a heavy chain, this is a heavy chain. Now these two heavy chains are connected by disulfide bonds, as well as the heavy chains and the light chains are connected by another disulfide bonds. Okay, now usually there are immunoglobulin A variant, D, okay, M, G, okay, E. So all these variants of immunoglobulin has some different biochemical structures. Now those structures are variable because of the change in this part of the molecule. This is called as the variable portion. Now the part of the molecule which is the tail part is called as a constant, which remains the same for every molecule. Okay, so usually when immunoglobulin acts, it usually helps in fighting with antigens. Okay, so what happens, or pathological antigens, so antigens come and attach to this variable portion of the immunoglobulin molecule. So this is called as the antigen binding site. Okay, the picture over here showing is, the picture shown over here is movement of neutrophil during the process of phagocytosis. So before phagocytosis, what happens, the neutrophil, whenever supposedly this is a viral or bacterial or any immunologically active foreign body is there inside your blood, what happens, the first line of defense cells that come into play is called as the neutrophils. Now this is showing you the movement of the neutrophil towards that pathogen. So first step, what happens, the neutrophils will be circulating in the blood vessels. So whenever it encounters or it faces this pathogen, whatever the pathogen emits some substances. Okay, those are called as chemotactic substances. Now these substances releases the chemicals which are detected by the neutrophil as foreign, and they start to increase its permeability through the membrane. Okay, so for that, what happens, the cells, the endothelial cells, there is increased permeability of the cells of the endothelial lining. Okay, what happens, the neutrophils start to stick to the sides of the endotheliums, and there is margination. This step is called margination. Okay, now as the cells come and marginate towards the endothelium, what happens, there are some events that is taking place which leads to increasing the space between the intercellular junction. Okay, so this step that is taking place over here is called as diapedesis. Okay, the neutrophil in fact tries to squeeze out from this cell, and when it squeezes out from the endothelial lining, it comes to the extracellular fluid. Now this chemotactic substance or source attracts this neutrophil towards the site. Okay, now the main event that is being shown over here is being elaborated in this picture. Okay, now in this picture, this figure is showing you migration of neutrophil from blood into inflamed tissues. Okay, supposedly this is the inflamed tissue over which a blood vessel is lining. Okay, now the neutrophils will be moving around the blood vessels. Now as soon as this blood vessel encounters to some pathogens, supposedly these are the not pathogens, like chemicals which are secreted by the chemotactic substance, there is release of some cytokines by the tissues that is in the vicinity. Now these are the cytokines supposedly. Now what happens to the cytokines? Now in the inflamed tissue, when cytokines are released by the tissues, the cells, the neutrophils, undergo some modification in the blood vessels. The first thing that happens is that in the blood vessel endothelium, there are some receptors. This Y-shaped structure that you can see is called as the selectin. This is called as the selectin. Okay, now what happens, this selectin adheres itself to the receptor which is present on the neutrophil cell membrane. Okay, so it gets attached, and because of that what happens, the neutrophil gets adhered. Okay, so because of that what happens, the first step is called as rolling adhesion. Okay, in this step what is happening over here, there is tight binding of the neutrophil is occurring over here. Okay, now as soon as the lectins are being binded with the receptors of the neutrophil, another receptor which is called as the intercellular adhesion molecules, ICAM1, this also gets adhered to the receptors of the neutrophil. Then what happens, the endothelial space between these two cells increases. Okay, and because of that what happens, the neutrophil is easily passing out from the gap. Okay, so this is called as diapetesis, and when it reaches the inflamed tissue, it migrates. This is called as the migration step. Okay, migration. Okay, this picture is showing you the structural unit or the functioning unit of a nephron. Okay, if you cut a section of the kidney, the ultrastructure that we'll get is the nephron. Now there are, in the kidney we have a medullary part and the cortex part. So if this is a kidney, okay, so the very center of the kidney is called as the medulla, and the outer portion is called as the cortex. Now this is showing you the cortex and this is showing you the medulla. Okay, now somewhere around here the cross section has been made and you are getting the tissue in this figure. Okay, so this is showing you this as the cortical nephron, which is closer to the cortex, and this is closer to the medulla. This is called as juxtamedullary nephrons. The obvious distinctive feature between these two nephrons is that one is longer than the other. Okay, then here in the nephron you can see we have the collecting duct which collects the filtrate which is passing from the nephron. Okay, and around the nephron you can see some vessels are encircling the whole nephron. This is called as the vasa recta. Okay, vasa recta. Vasa recta consists of the vessels which is coming from the interlobular artery and the veins. Okay, which in fact is coming from the arcuate artery and vein, which in fact is coming from interlobular artery and vein. Okay, then the glomerulus, we know the nephron is divided into some parts. Okay, the part which is a cup shaped is called as the Bowman's capsule. Okay, attached to that is the proximal tubule, proximal convoluted tubule. Then we have the loop of Henle, which is a hairpin loop of Henle. Then we have the distal convoluted loop, and we have the then collecting duct. Okay, so here in the Bowman's capsule we have some fibers, tuft of vessels. These are called as the glomerulus. Okay, now it is because of the efferent and the afferent arterioles. Okay, now the many such collecting duct joins together and forms the duct of Bernoulli. Now what is this figure showing you? This is showing you the typical respiratory membrane of lung. Okay, so it is shown along with the structure. This is a biconcave structure called as the RBC, red blood cell. Okay, so this is the lung area. This is where the lung is there, and this is where the blood vessels or the pulmonary vessels are there. Okay, supposedly this is the lung. Okay, connected with the trachea and the bronchus, and around the lung we have some vessels called as the pulmonary vessels. Okay, now somewhere around here the cross section has been made and we have kept it over here. Okay, so this is the lung tissue. We have the vessels over here and intermediate areas. Okay, so coming from inside the lung to the outside, what are the layers? So this is, so if we look into the lung, okay, we know the lung is the terminal. There is a respiratory bronchioles was there. So in the respiratory bronchioles, it is connected with the alveolar ducts. Now inside this alveolar ducts we have single alveolus. Now the alveolus are lined by some fluid material called as the surfactant. Okay, so this is a surfactant for you, the inside of the alveolus, which is attached to epithelial lining of the alveolus. This is the epithelial tissue cell nucleus. Okay, attached to any cell we know there's a membrane. Okay, so this is the membrane for you. Now on the other side, okay, we have the blood vessels. Okay, similarly we know the inner lining of the blood vessel is made with endothelium. So this is the nucleus of the endothelial cell. This is another nucleus of the endothelial cell. Okay, it is connected like this. Okay, now also this is a cell, so that's why it also should be capillary basement membrane. Now in between these two we have the interstitial space. So this is the respiratory membrane for you, with the help of which the oxygen is diffused from the lung alveolus to the RBCs, as well as the carbon dioxide from the RBCs to the lung alveolus. Shown over here is a cystometrogram. Okay, a normal cystometrogram showing the micturition contractions and reflexes by the dashed lines. Okay, the x-axis is showing you the volume of urine that is getting accumulated and the gradual increase in the pressure. Now in the urinary bladder there is a reflex pathway. Now we know the reflex consists of a central analyzing organ, okay, which is connected by a sensory arc. Okay, then we have the motor arc, okay, to the target organ. Okay, the sensory arc is connected to the sensory receptor. So similarly, in case of the urinary bladder, the sensory receptors are the stress receptors that are present in the muscles of the body of the urinary bladder, especially the detrusor muscles. Okay, the sensory arc are nothing but the sacral nerves which are coming from the detrusor muscles to the central relay station, that is the sacral plexus, or the neurons that is present in the spinal cord. Now from the spinal cord, the motor arc, it consists of the parasympathetic fibers, which are again going back to the same target organs, that is the body of the urinary bladder, specifically the detrusor and the posterior urethra. Now this is the reflex arc which comes into play in urinary bladder. Now what happens? The kidney does not eject the urine in one shot. It emits, or it proceeds just few milliliter at times. 20 to 30 ml of urine is passed from the kidneys via the ureter to the urinary bladder. Now what happens, now as it is increased getting inside this bladder, now at the beginning when there is no urine in the bladder, there is the pressure that is equal to 0 centimeter of water, 0 centimeter of water. Okay, so there is no pressure being there when there is no urine. Now as soon as few milliliter of volume of like 50 up till 50 ml of urine is passing inside, what happens, the pressure gradually rises from 5 then 10 centimeter of pressure. Now why this is happening, because when the urine comes from the urethra, the presence of the fluid volume, it increases the pressure inside the detrus in the bladder, because there is tonic contraction of the muscles of the body of the urinary bladder. Now this constant pressure that is been creating over here by the muscle, as well as the presence of fluid or urine inside the urinary bladder, it leads to the genesis of the reflex arc. Now for this reflex arc will take the sensation with the help of the stress receptors, sacral nerves, then go to the spinal plexuses, then it again comes back by the help of the parasympathetic, and again gives the signal to try to evacuate the very little amount what is accumulated in the urinary bladder, to evacuate that. But that is not possible, because simultaneously the external sphincters are also being contracted, so it is not being able to overcome the pressure that is being created in the external sphincter. So what happens, it comes and goes back. Now this step, that is increasing the volume of the urine in the bladder and increasing the pressure, will keep on increasing as the volume will increase, and this cycle will repeat after every few minutes to few hours. A reflex arc will arrive, and that will lead to some contractions, okay, till it reaches a level of 300 milliliter, then after which it repeats after every hour, okay, till the pressure exceeds the external sphincter's pressure, as well as the contraction that is been present because of the voluntary stimulation of the external sphincters. Now as soon as it overcomes that pressure, what happens, the pressure drastically decreases and it goes back to the 0 centimeter water. This is called as the cystometrogram, and recording of the cystometrogram helps you detect any abnormalities in the spinal reflex pathway or in the micturition reflex pathway.